Next one, number 7, telomere. So telomere normally comes at the ends of the condensed chromatin, of course, which forms chromosomes. So at the end of each sister chromatid, we actually have telomeres, which are made of repeating nucleotides that do not form. So telomere, repeating nucleotides, repeating nucleotides that do not form genes, that do not form genes. So what is their function? They prevent the end of the chromosome from accidentally attaching to each other. They prevent the ends of chromosomes from accidentally attaching to each other. From accidentally attaching to each other. So they prevent it from attaching to each other. And they help prevent the loss of genes also. And they help prevent the loss of genes, of course, during division. So, of course, a section of nucleotide is normally lost when a new DNA molecule is copied. So it is important that these nucleotides that are lost are telomeres and not the genes themselves. So we shouldn't lose the genes, because losing the genes can be fatal to the organism. That is why telomeres are there to prevent this problem. So we go to the next question, number 8. Reading toolbox, word origin. The prefix pro means earlier than or prior to. Explain how this meaning relates to the word profess. Explain how this meaning relates to the word profess. So you are supposed to explain this. So telophase is the end of mitosis. Telophase is the end of mitosis. So when we are saying earlier or prior to, okay, we are talking about prophase. I mean number 8. This is number. The prefix pro means earlier than or prior to. earlier than or prior to. So actually when we are looking at prophase, not telophase, prophase is actually the beginning of mitosis, is the beginning of mitosis, or the beginning of nuclear division. The beginning of nuclear division. Then telo means distant, far, end. How does this meaning relate to the words telophase and telomere? So telophase is the end of mitosis. Telophase is the end of mitosis or the nuclear division, while telomere actually is the end of the chromatid. The telomere, as we saw it, is at the end of the chromatid. So at the end of the chromatid, telophase, the end of mitosis, and telomere is at the end of the chromatid. Prophase, on the other hand, is actually the beginning of mitosis. So they expect you to express yourself in terms of what you know. So these questions are actually meant to see if you really understand what the words mean. Question number 10. The term mitosis comes from the Greek roots mitos, which means thread. How does this meaning relate to the process of mitosis? How does this mean or relate to the process of mitosis? So this means division of the DNA. So the DNA strand actually is first spaghetti-like or string-like. So when we are talking of mitosis, meaning thread-like, it means separating these thread-like structures, because at first, mitosis actually, when people saw it first, they were not seeing all the details, but they were seeing these amazing threads which divide into two. So this means division of the DNA strand, or precisely chromosomes. So let's go to number 11. Number 11, The cell cycle has four main stages: G1, S, G2, and M. What occurs in the cell during each stage? So we already discussed this, so we will go direct into a summary of each. G1, there is increase in size, replicates organelles, replication of organelles. So organelles are replicated here. G1 checkpoint, we have G1 checkpoint. There is G1 checkpoint. Make sure the cell is getting big enough to divide. Make sure the cell is big enough to divide. Signals other cells to see if division is necessary. Signals other cells to see if division is necessary. To see if division is necessary. Where cells spends most of their time. Where cells spend most of their time. So cells actually spend most of their time here. Then we have G2, S phase first, synthesis phase, DNA replication, or DNA division here, replication or synthesis. Then we have G2. What happens during G2? So you can pause your video and note them. Note the things that happen here. There is more growth. There is also G2 checkpoint. Make sure that the DNA is undamaged. Make sure the DNA is undamaged. Checking also the size. checking that the need for cell division is still there. Checking that the need of cell division is still there. So at any time here, still cell division may be stopped. So is everything there in order to divide? So in animal cells, the centrioles replicate. Is everything there in order to divide? In animal cells, the centrioles replicate. So the centrioles will replicate. Then we go to the last and final phase, that is the M phase. The M phase. There is M checkpoint. So these checkpoints are actually very important. Makes sure chromosomes are properly attached. Properly attached. Happens in the middle of mitosis. This process here happens in the middle of mitosis. So mitosis is the division of the nucleus. During mitosis is the division of the nucleus. The division of the nucleus and its contents. And its content. So chromosomes are separated and distributed into two new cells. Chromosomes are distributed into two new cells or daughter cells. So we'll stop here for this. Then in the next video we'll be looking at more of what actually happens during mitosis.